The 2012 Albert Lasker Basic Medical Research Award honors Michael Sheetz, James Spudich, and Ronald Vale for discoveries concerning cytoskeletal motor proteins, machines that move cargoes within cells, contract muscles, and enable cell movements.
If it weren't for the incredible experiments of Hugh Huxley, none of this would have happened. He is for sure the person whose shoulders that not only I, but Mike and Ron and others stand on.
Intrigued by muscle motility from his postdoc experiments at the Huxley Lab, Jim Spudich applies his training as a biochemist to understand movement by reconstituting it from purified components.
And there weren't any in vitro systems for that. So it was a no-brainer, given that background, to want to start my lab trying to find a way to develop an in vitro motility assay.
Hey, Simon.
When Mike Sheetz arrives to spend his sabbatical year in Spudich's lab, they decide to try the in vitro assay using the alga Nutella.
Okay, so this is Nutella. It's a freshwater pond organism. And the amazing thing about it is that the cells are very large.
And the Nutella cells were indeed, in some cases, 3 inches long and 1 millimeter or 2 in diameter.
As soon as we cut open the Nutella and laid it out and put the myosin-coated beads on,
and they come in contact with the actin cables and then start moving in a unidirectional fashion along the actin.
They moved in the very first experiment, and it was just terrific.
Well, he was ecstatic.
And this was for sure one of those eureka moments. Mike came running into my office saying, "You got to see this." It worked the very first time because it was meant to work. But you can see that you get quite convincing movement of these things chugging along these tracks. And this was the first quantitative in vitro motility assay.
So Ron was a graduate student there working on nerve growth factor. We were talking about membrane protein dynamics and these sorts of issues. And I told him about what we had been doing with this in vitro motility assay.
Sheetz and Vale wonder if nerve cells are using the same motion-generating machinery and decide to use the squid with its giant axon for their research organism.
That was 1983, the year of El Nino, which was a meteorological phenomena that warmed the oceans around California, and the squid mysteriously disappeared from the coast of California.
They head off to the marine biological laboratories at Woods Hole, where squid are plentiful, and where they find a new type of microscopy being developed with video cameras and computer enhancements.
So it was a very exciting new technology to merge with a field that I knew very well, which was biochemistry.
Their vision of finding an actin-myosin-based system is rapidly dispelled, and they decide to use an approach of reconstituting movement from purified components.
So the first step in trying to dissect the mechanism was to break apart the interior of the squid giant axon and study it on a microscope slide.
And what was evident when we looked at this in the electron microscope was that this quote-unquote transport filament was only a microtubule. And so that simplified our life quite a bit.
Sheetz and Vale add purified organelles and ATP to pure microtubules with little success. When they add soluble protein from the cytoplasm, the unexpected happens.
So this was the eureka moment that happened at 2 o'clock in the morning when we couldn't believe our eyes. Looked at the video microscope and saw something completely surprising and unexpected. In what was supposed to be a simple control experiment in which we weren't supposed to see any movement, the tracks themselves, these microtubules, were gliding, translocating along the surface of this glass cover slip. And we then realized what the magic ingredient was in this protein mixture that was making the organelles move. It was a protein that was free in the protein mixture that was sticking onto the surface of the glass cover slip and propelling these microtubules to move across the glass. And we then realized that this was the kind of simple assay to study and dissect motility that we've been looking for.
And that initial study showed that the kinesin molecules were walking along a single, what is called protofilament in the microtubule. It's made up of 15 or so individual paths of tubulin dimers, and the kinesin was walking in a hand-over-hand fashion along one of those.
So it was a completely new type of molecular motor. So that was very exciting to discover something completely new.
These three scientists continue to develop robust experimental systems, expanding the growing field of molecular motors through their in vitro assays and other research activities, as well as elucidating the potentials for medically targeted applications.